Hola a todos, buen día y bienvenidos a la semana 3 en la clase Microbiology 205 lecture. Welcome to week 3 of microbiology lecture and your journey into the world of the unseen. Now, it can seem strange for many to think of microorganisms and especially unicellular bacteria as living organisms, which are at least on one level no different from humans. The very basic biological purpose of the human is to propagate the species, just like with any other organism. Likewise, the very basic biological purpose of a bacterium is to propagate the species. So bacteria respire, they don't breathe in and out like we do, but they respire, they exchange oxygen and carbon dioxide with their environment, they respire, they ingest nutrients, they metabolize those nutrients, they excrete waste products, and they reproduce just like humans. The point is, in order to do all of that living stuff, bacteria need genes just like people do. Now, bacteria carry their genes on a single circular chromosome. Humans, we have 23 and they're linear. Well, we have 2 pairs of 23, 46 total, but they're linear. And so this week we take a look at microbe metabolism and bacterial genetics, and we'll examine first how bacteria can manufacture the energy necessary to do all of that living stuff, and second, how they can transfer characteristics, like, for example, antibiotic resistance, to their offspring. We call offspring of any organism the progeny. So I have provided links to 3 short videos to help you understand the concepts here in week 3. The first is a video describing the process of aerobic and anaerobic respiration. More on that in a minute. The second explains a central dogma. Central dogma is the flow of genetic information from DNA to RNA to protein. And the third explains the methods of gene transfer between bacteria. The question is, if as you learned last week, bacteria reproduce via binary fission, and the daughter cells of the parent cell. The parent cell yields 2 daughter cells and their genetic clones. So wherein does the genetic variability in bacteria come from? Because if there's no genetic variation in bacteria, then those species will die off. That comes from transfer of genes between bacteria. Some bacteria pick up other genes from the environment. Bacteria can transfer genes between them, between members of the same species, and even between members of different species via bacteriophages. So you'll learn more about that in this week. Okay. So chapter 6 is microbial metabolism. That's all about energy. The penultimate source of energy for all of life is the sun, including bacteria. You are, I'm certain, with the photosynthesis. If not the process, at least you know that's where plants absorb light from the sun and they turn it into chemical energy, and that's how plants live and survive and grow. Photosynthesis is that process whereby plants and some other organisms, like there are photosynthetic bacteria and, of course, algae, which absorb light from the sun and convert that into chemical energy, which is used by the rest of us. So for instance, plants absorb light from the sun, convert that into chemical energy and make glucose, and then we eat the plants, and then that energy is transferred to us. Or a rabbit eats grass, and that energy is transferred to the rabbit, and then a fox comes along and eats the rabbit, and then that energy gets transferred to the fox. That's the way it works in the natural world. So we call that energy ATP, adenosine triphosphate, ATP for short. And you'll learn how bacteria are able to harness energy, make ATP, and do their living thing, right? I have posted a video explaining the process of making ATP in bacteria, and I again, I have some more to say about that here in a minute. Well, let me say it right now, otherwise I'll forget. Looking at the quiz results from last week, um, not as well overall as the quiz from week 1, but in the material here in week 3 with regards to energy and energy production can get a little fuzzy maybe, or a little, I don't want to say difficult, but a little challenging to keep all the ones, twos, and threes apart, or A, B's, and C's. So I have a way that I'm hopefully going to help you with that. I have um, some infographics that I created for another class that I'll share with you that will hopefully help you understand this process of respiration. Okay, chapter 7 is a blueprint of life. Your genome is the sum total of all the genes that make you who you are. Everything that you do, think, feel is controlled by your genes. Now, our genomes are wrapped up, literally wrapped up, in those things we call chromosomes, and our chromosomes are made of DNA. So are bacterial chromosomes. Now, the central dogma describes how those genes in DNA, double stranded DNA, are transferred, converted into the instructions in RNA, single stranded RNA, to make protein. Everything that you are, everything about you is protein. When I say protein, don't think of like hamburgers and, and steak on the grill, right? Think of proteins as chemical entities that um, that are structural. For instance, building cells, that are functional. For instance, hemoglobin is a protein that carries oxygen through your blood, right, from your lungs to your tissue. So everything that happens within the living organism is protein or controlled by protein, right? So think of protein that way, not meat. And and so the central dogma describes how those genes are converted into the message RNA, which makes the protein then, or directs the making of the protein. It's called the central dogma. And so you learn about DNA and RNA, genes and proteins. Now the central dogma is true, it's universal for both eukaryotes like humans and prokaryotes like bacteria. Okay, and then finally in chapter 8, there's bacterial genetics, as we humans try to maintain stability and familiarity within our environments. The fact is that our environments are always in flux, they're always changing, right? And some of those changes are obvious, like here in the East, it was 24 degrees, I think, when I woke up this morning. It's probably in the in the high 30s, low 40s right now. Those are obvious changes in the environment, but still things change. Moisture um, level changes, and so our environments are always in flux, even though we may not think about it that way. Well, for a bacterium, they're always in flux as well. We sometimes notice the changes if they're big enough, like a natural disaster, for instance. But we are constantly, our bodies, our systems are constantly adjusting to maintain, we say homeostasis, or to maintain an even, level, calm environment within based on the changing environment without. Well, microorganisms are no different. Bacteria must constantly adjust in order to survive. They're continually changing environments as well. So you learn about bacterial mutations, which is mutations as a fuel for natural selection, and how bacteria evolve, and very rapidly, I might add, to survive and reproduce in their ever changing environments. All right, housekeeping. Make a discussion post early in the week so you can fully engage in the discussion. I have made that comment to several of you who waited until Friday evening to make your initial post. You still obviously still get the credit because that's the rule. But if you post earlier in the week, then that gives your peers enough time to read your post, digest it, do a little bit of research, and respond properly back to you. So the earlier you post your initial reaction to the prompt, the better off you are with regard to your peers and the whole discussion process. Okay, your initial post is due no later than Friday. That's this Friday, January 23rd, 11:59 p.m. GCU time. There is a microbial metabolism and genetics assignment or homework, and microbial metabolism and genetics quiz. Both of those are due on Sunday, no later than Sunday, January 25, 11:59 p.m. GCU time. Check your class resources, as I'm always adding documents, various resources in there. And I probably will post those infographics there as well. But I usually, if I put something there, I'll make an announcement. I'll post an announcement and say there's a new resource to check out. All right. Pay particular attention to due dates on all assignments. Proper credit can only be given for complete and timely submissions. Okay. Any questions or issues along the way, contact me as soon as possible, preferably. You can message me directly, 804-229-7993. You can use your Halo message box or inbox. Those are the two quickest ways to get a hold of me. And yes, certainly you can use campus email. If you message me directly, make sure you include your first and last name and tell me the class that you're in, Microbiology 205 Lab or Microbiology 205 Lecture, because I'm doing both. Okay, that is it, I think, for now. Let me know what's going on. Have a good rest of the week. Oh, and if I will be updating your grade books. If you see in your grade book there's a big red overdue. I explained this in my announcement last week, but if you see a big red overdue there, don't freak out. If you know you did your assignment, then everything is good. I just have to manually transfer grades from Connect over to Halo, and I'll usually do that on Monday. Since I'm 2 hours ahead of Arizona, I'm usually not up at 2 o'clock in the morning transferring grades. Although I was last night, so those grades are updated in your book, but your participation grades are not updated. I'll do that today. Right? Let me know what's going on. Have a great rest